Can everyone see the screen?
Yeah.
Stop the beginning. Sorry about that.
Wow.
Can everyone see that? Yeah.
Yeah.
Yes. Okay.
Bless me.
While we're waiting for Margaret to join us, I'll just go through a couple of points from last week. They're about tepals, so I promise I won't spend more than 5 minutes because you're probably sick of hearing about them, but just because the questions were raised last week. The first one is just to show you in the hellebore, in the buttercup family, we talked about how the outer ring of tepals was coloured like petals, but the petals themselves have been converted into these horn-shaped structures called nectaries, which obviously make the nectar for the insects to attract them to the flower. So I know some of you have been looking at the hellebores this week. It's quite a good time to inspect them. So basically, that's the nectaries, which would be petals in most of the flowering plant families. But in the case of some of the members of the buttercup family, they have these various adaptations. So don't always look like that. Okay. And the next point was, don't get too bogged down in this. It's just someone asked me about the origins of the tepals. And basically, it's the perianth, or the petals and sepals, developed from things called primordia, which are just growth regions in the embryo. And they're controlled by a series of genes called the Madsbox, or A, B, C, E genes. And basically, to put it simply, the number of genes and their combinations results in 3 different options, as far as I can see. The most primitive one is in the Magnolia family, which are a very early family in the flowering plant kingdom, and they just have 1 whorl of tepals. So certain genes are switched on for that. Most flowering plants have sepals and petals in 2 whorls. A different combination of ABC genes are switched on for that. And finally, the one that was puzzling us was in the lily family, the tepals there, which are 2 whorls of tepals, originate from a single whorl of sepals, as far as I can tell, which are from a different combination of genes. So it's quite complicated, and you can't tell by looking. And the Q Plant Glossary, which is a very good book and definitely to recommend, says tepal, a division of the perianth, i.e. a sepal or a petal, used especially when it is unclear which it is. And I think that's a great definition. So let's forget about the rest and stick to what Q Glossary says. Okay, so that's where we are. So are we all here yet, Tricia?
I believe we're now. Margaret, are you with us? Margaret?
Lizzie, while we're waiting, did you by any chance record last week's?
Yes, we did.
Is it possible to send me a copy, because I couldn't make it?
Right. If you send me an email, then I'll send you the link back again.
Brilliant. Thank you, Tricia.
Where's Margaret gone?
We've sent some documentation out to back up this week's talk, but I know that I think it was my fault with the way I saved the PDF. It hasn't made it very easy to transfer. So if there's still a problem, you know, we'll have another go. Tricia sent it tonight, so it should be okay. If you've got a problem, we'll have another look at it. Okay.
Margaret, are you there?
Yes, can you
Okay.
hear
Yes.
me?
Yes,
Right.
we can hear you. We can't see you yet.
Is that any better?
No, it's
No.
somewhere on your screen. It'll say start. There you are. We've got the top of your head.
Yep, right.
That's brilliant, Margaret. We've got you now.
Okay, so it starts. Yeah, are we ready?
Sorry, sorry I wasn't there. Has Lizzie started?
No, she hasn't started this evening, Margaret. She's answered some questions on sepals and tepals from last week.
Ah, good.
So we've answered the question. We've solved sepals and tepals as per what Q say. So it's over to you to introduce Lizzie, or we can go straight to Lizzie.
Shall we just go straight through?
Right, I am going to mute everybody, and then Lizzie, can you unmute yourself?
Right.
Okay.
I'll have to find myself. I haven't got on there now.
Have you got your mute? Right.
Hang on, I just can't find myself.
Right, you're still on mute now.
Right.
That's back. Right. Okay. And so over to you, Lizzie.
Okay. Well, tonight I'm going to talk about the daisy family, the Asteraceae, possibly another family. We'll see how we get on with this one. But the daisy family is a really large family of species, including a lot of very familiar plants like the dandelions, the daisies, the thistles. And it's one of the largest families in the British Isles. They're mainly annuals or perennials, and they're mainly herbaceous plants. So that means they're not woody. They tend to grow in the spring, grow vegetatively, develop their flowers and seeds, and then die back. If they're annuals, they'll set seed and grow the next year from seed. If they're perennials, they'll just grow back again in the spring. So we're going to mainly concentrate on this flower structure of the daisy family, but I just want to talk about the leaves a little bit because they are very important. And when you get out in the field soon, those of you who are coming out on the field trips, you'll see that they are really important in identifying some of the more challenging members of the daisy family. So there's quite a lot of variation in the structure. You can see our well-known dandelion here, and the leaves can be completely entire. That means they're not serrated, they're not toothed, they're just smooth. They can be lobed, they can be pinnatified like these ones, where they're divided up into little indents like that. They're mainly alternate, sometimes they're opposite, and the leaves are either have stalks or not, and whether there's leaves on the stem or not is also very important in determining some of the different species. So there's another example. If Richard's on, he'll recognise that. It's his favourite plant, that's Crepus mollis. And as you see, this one has leaves on the stem, if I can get my cursor to work, and they alternate up the stem, and they help to identify that this belongs to the genus called Crepus mollis. I've forgotten its English name, sorry. Hawksbeard, maybe not. And there's a thistle, another member of this group, very different leaves again. So that's just a few examples to show us that there's such variation already in these species. So let's have a look at the flower heads. Now, I've picked this group this week because they're so different to the flowers we've looked at so far. Basically, what looks like a flower, like a dandelion flower, is actually a packed head of tiny flowers called florets. And the head or capitulum, they're all inserted onto this head, this disc-like receptacle, and here it is. Now, each of those is a tiny little flower called a floret there, and you can just see the stigmas coming out, those little curly structures there. I'll come on to them in a minute. That's a close-up, just showing you that they look different in the middle, but they're just the same. They're just not quite out yet, so they're all curled in like that. And this head has, this capitulum head with all of these flowers in it, has this surrounding ring of bracts. This is called the involucre, and these are called the filaries. But the word to learn is the involucre. That's the important one here. Now, inside here, the tiny florets can vary. They can either be tubular or ligulate or both. And I'm going to show you what those differences are. But just remember, they're all tiny flowers packed together in that one head. And of course, when the flower sets seed, you get the characteristic head, which I'm showing you because when the seeds go off, it shows you the capitulum where the florets would sit. Now, this isn't a dandelion one. I haven't been able to match everything in terms of the different stages of development of the flowers because I just haven't got the photographs basically. But you can see the tiny holes. These are where the little fruits or the achenes sit. The florets sit in there, and that's the involucre all flattened out. So you've got this information that I've sent you, and all of this information on here is going to be on the next diagram. So let's just go to them now. So this is a diagram of the head of the daisy, just like you've been looking at with the dandelion there. And you've got ray florets, and this particular, sorry, ray florets here and disc or tubular florets there on this head with the involucres here. So I've just given you a diagrammatic representation of what could be on the head. You might get just ligulate or ray florets, just disc or tubular, or one or the other, as you're going to see in a minute. Now, just remember that this whole flower that I showed you last week is packed into one of these. So everything in there has to be in here in some form. So there's the sepals that we've been talking about so much, and they have become this pappus of hairs. Now, most, but not all of the daisy family do have a pappus of hairs, but some don't have anything there. Then you've got your ovary down here, inside this sleeve, which is called the corolla tube. So the petals here have all been turned into this tiny tube, which is closed at the bottom, then opens out into this, what's called a ligule or tongue here. And then down here, you've got that's the top of the stigma, and the style goes down here to the ovary, and those stamens are all attached in a tube around here. So all of that flower is condensed into that tiny floret. Is nature not amazing? So I've labeled them on here for you to show you. So there's the corolla, the petals, which are all joined together. Then you've got your stamens on this tube. The stigma, style, and ovary, which is the carpel, is this part here. And then the calyx, or pappus of hairs. And I've sent you a diagram, this one. It's not perfect because I drew it ages ago just to go out with the practical. But it just gives all the labels on here and relates them to the photograph. So you can have a look at that yourselves. I think I've sent you that one anyway.
Yes, you have.
Yeah, oh, good. Okay, so now if you look at a head here, this is a more opened out head, just now that you've had a look at those diagrams, and you can see the curls, stigma, style, and ovary, and the anthers attached there, and all the way around here. This one's dying back now. It's all collapsing outwards, and all of those tiny flowers are quite visible on there. And that's a lovely one to show them closer up. And really just to show you here that, yes.
Can you hear me?
Yes.
I understood that the anthers are touching, they're attached onto the wall of the corolla tube.
That's right, yes.
The anthers are touching, and as the closed stigma pushes up, as it grows, it pushes the pollen out at the top.
Yes.
Yes.
Yeah.
So that the pollen is pushed out before the stigmatic surfaces of the inside of the curve of each branch is open.
Yes.
Okay.
That's very good. No, it's a really important point because that's stops self-pollination. And so what Mark is talking about there is the fact that the stigma grows through and pushes the pollen up. It's a really important point, and we'll come back to that in a minute. Thank you, Margaret. I was just really to show you the florets, and there's the seeds as they've developed from the bottom of, that's the bottom bit here coming off. Now, the other florets are tubular, and the knapweed is a good example of this one. And you can see the tubular florets here. So this is knapweed, which you possibly all recognise, I hope. And we see plenty of that around up at Teesdale. And that's just a section across to try and show, this one was dying back when we cut it open, but you can see the tubular florets, can't you, here? And the bits of the pappus of hairs. And of course, this is the capitulum again there. And it was quite hard to get these out. I couldn't dissect them very well because the plants were going back. But that shows you, that shows you the tubular floret there. There's the corolla tube. There's the anthers. And as Margaret says, the stigma is going through there up to the top. and that's the seeds. See, they're all quite, the seeds are really important actually in identifying the species. They all have different patterns on them. And that one, just again, I sent these diagrams to you. Oh, sorry. Oops, here, and most people just recognize tubular and ray florets, but in the Streeter book, the Flora, the quite recent one, I've forgotten its name, they use both of these, but I don't think you should worry too much about that. They're just variations of the same idea, and that's a better example of the tubular floret, like that one there. Now I've used Colesford here because it's out now and you can see it. I had some nice photographs of it anyway, and it shows you a species where both ligulate and tubular florets are present in the same flower. So we've got outer ligulate florets, and they don't have stamens, so they're essentially female. Inner tubular florets can only make pollen, although they do have a bit of, a remnant of ovary. It doesn't develop, so they're effectively male. And this is the point that Margaret was making earlier, really, about trying to encourage cross-pollination, because if the flowers mature at different times, it prevents the pollen from fertilizing itself. It's much better if the genes, we said last week, can be transferred to a different flower to cause, sharing, crossing the genetics and increasing the genetic diversity. But of course, if there isn't any fertilization, there's usually a little window at the end of the season where self-pollination can take place. So here's a Colesford flower. I think these are so pretty. And you've got the outer ligulate florets here, which are really very fine. And remember, they're female. They've only got the ovaries. And then these little tubular florets in the middle that are essentially male. They only make the pollen. And I've mentioned that already about the difference in maturation. I'll send you that diagram just to show the different bits, that just shows you the bud of the Colesford with these characteristic triangular bracts. And this is a section through the Colesford to show you the ligulate flowers and the tubular ones in the middle. And then I've drawn one of each here again. Not great diagrams for the show, but I want to show. So you see how much finer they are than the dandelion ones, Colesford, but you can see you can still see the ovary, the pappus of hairs, the corolla tube, and there's the anthers, and that's the emergent stigma. I think these ones are just really fascinating, these little tubular flowers in the middle. And you can see the corolla there, and then the anthers and the stigma there, I think. Oh no, actually, it would just be anthers because it isn't a stigma there, actually. Let's just see that. So my diagram's in the way, my little thing about me is in the way, I can't move it. I'll see on the next diagram. That's better, yeah. So here you can see the central tubular flowers quite clearly, and the outer female ones. Now, the other thing is that because you have ligulate flowers like the dandelion, tubular ones like thistles, you get different types of flower head. So as well as getting florets called ligulate, tubular, and disc, ligulate and tubular, I beg your pardon, because you get different combinations, you end up with different flower heads. So you've got a ligulate flower head, a disc or tubular flower head, and a radiate flower head. So because you get different combinations of the florets, you get different types of flower head, and they're obviously really important in classifying the different species within the daisy family. And we've looked at the Colesford, that was radiate. Here's another one, which is leopard's bane, and there's your flower head. A lot more tubular florets in the middle, and you can see they're just maturing around the edge. These are still tubular, and then you've got your ligulate flowers on the outside. And the daisy, again, another radiate one, just tubular florets in the middle and ray on the outside, and that's, you can just see them starting to mature in the middle there. The ligulate ones aren't very clear. I couldn't get any really good photographs of the ligulate ones, to be honest. And that just shows you them starting to open out a bit more. And finally, the disc thistles or related members, species related to the thistle family. These two here are thistles, and you can see their tubular flowers there. And that's just another ligulate head. That one's the dandelion, and this one, of course, you're recognised by the leaves here, is a sow thistle. And that head's rather beautiful in its symmetry, isn't it? You can see all the little stigmas coming out there. So that's all the same type of ligulate flower. Now, to finish this section, I want to look at another radiate flower, which is the yarrow, because it looks so different from something like a daisy or the leopard's bane that we looked at there, because this has got both ligulate and tubular florets, but they're just tiny. So there's the whole head there. But if you take the flowers, you can see that that's the equivalent of the daisy flower. So you've got your tubular florets in the middle, and then your ligulate ones on the outside, but they're so small and they have far fewer numbers of florets within each flower. And that's just a close-up really to show that. And you can just see the structure there. I tried to get it when it was a seed, but I couldn't get that to work very well. And that's just a more mature head on a whiter milfoil flower, same species. So that's the main structure. So you've got radiate, ligulate, and then you've got tubular. And some more examples just generally of species you might come across in this family are the burdock, goldenrod, which we see on by the riverbank up in along Bilo Force, Mountain Everlasting, of course, which we see hopefully plenty of on Teesdale. And that's the end of the daisy family. So shall we pause there, Tricia, and see if anybody's got any questions or any comments to make about that? Can we do that?
Yeah, I believe we can. Yeah, my touch screen's not working very well at the moment. Does anybody have any questions?
Yes,
Can you hear
please.
me? Yeah, go on, Margaret.
Oh, so sorry. Lizzie, you didn't say much about the fruits of the papas.
No,
You showed the young dandelion. I think I'm right that the dandelion then, to which part? Yes, part of the fruit extends into the stalk and your papas opens out, each one into this ray-like top. And then they tend to meet around each other, giving you the usual dandelion clock. But there's quite a lot of variation between whether they've got the papas sitting immediately at the top of the fruit, or whether the papas is on a stalk. And in some cases, I've just forgotten the species, but in one of them, it doesn't have a papas at all. It's got a little ring of scales.
Yeah, that's good. Laps,
But
is it
I
lap?
just wanted to mention there is quite a bit of variation.
No, that was really interesting, Margaret. I didn't know that actually about that. So that's something else you can look up and find out more about, the fact that there's such variation in the formation of the papas of hairs. That's very interesting. Thank you, Margaret. Has anybody else got any questions? Or anybody wants to share anything or add anything?
haven't got any questions, but there's a website that has all these really nice close-up pictures of gerbera daisies, and they do other families. It's a Canadian website, but they have lots of really nice close-ups, and I could send you a link in case any of the group would like to see those photographs.
Thank you, man. If you send it to Tricia, she's got the list. She doesn't mind. Yeah,
do
that's
that.
great. Thank you. Anybody else got any comments?
Just while we've got Margaret's expertise, since she's so good on the rose family, we've got Apomixis in the Rosaceae and Asteraceae. So is there any link between, or any reason that method of reproduction or clonal reproductions occurs in those two families, Margaret, particularly?
Can you repeat it, Richard?
Yeah.
Margaret, are you there? I've lost Margaret. Margaret's not saying anything.
You might be on mute.
She's muted.
Oh, Margaret's muted. Margaret?
Just a minute.
Got
Right,
you.
Okay,
I'm
Margaret.
here. Sorry.
Richard,
Hello.
hello,
It was-
Margaret. You're still muted. Oh, you're
No,
back
She's
muted.
not, Tricia.
I
Right.
was. May I finish? Why the
Sorry.
I was trying to send a message in chat to say that daisy, the fruits of daisy have got scales instead of a pappus.
All right. What's going on with that?
Now, did someone ask me a question?
I did. It was this, Margaret. And
What
it's, is there any reason that the Asteraceae and the daisies and the rose family have apomixis and it's not particularly widespread? So clonal reproduction.
I think actually apomixis is very much, sorry, let me explain to those who don't know what such a difficult word as apomixis means. It means that a plant can produce seeds or fruits without the need for fertilization. And it is much more widespread than is generally known, but there are some families in which it's more frequent than others. But I think it is probably a bit complicated to get into in this introductory course to flowers. I'd like us to go back to Lizzie.
Okay. Any more questions? Thank you, Margaret, and thank you, Richard. Any more questions before we go on to the next family? Okay, I'll just find them. Sorry.
Oh,
I've just lost me. I'll go. Now, the reason why I put this family in next is because they're another family with species with a lot of small flowers clustered together. A very different way of doing it, very different structure, but, I think it's worth just discussing it a little bit as we've gone through the daisy family. And this is the carrot family, Apiaceae. We're not too worried about the Latin names, but just mainly trying to get some idea about the variation in these important families. Now, this family is quite big as well and got quite a lot of variation in the species. Some of the species are things like carrot, which are obviously vegetables, and of economic importance, parsnip, celery. There's herbs like caraway and coriander. And there's quite a lot of common plants in this group, like pignut and the wood sanicle, which should be coming out soon, angelica and cow parsley. There's some nice sweet smelling species in this group, like dill and sweet cicely, and a few poisonous ones like the water dropworts and hemlock. So it's quite a range of interesting species within this group. And that's a photograph of sweet cicely in flower. I should say that the photographs are, a lot of them are by Geoffrey Chater and by my husband. And I'm very lucky that I've got these two, access to these photographs, because it makes such a difference to the talk. So this is hemlock to show you what it looks like, so you don't try and pick it. It's got this very characteristic blotchy stem, you know, which is really characteristic for hemlock. And oh, hang on, yeah, and yeah, so there's a few examples to start with. Now, the old name for this family was the Umbelliferae, and this is because the flowers have a very characteristic flowering head called an umbel. If you think about umbrella, it's like it makes a little mini umbrella. And each of those clusters of flowers is on a ray stalk, like this. So you've got this umbel-shaped inflorescence where all the branches come from the same point, like the spokes of an umbrella. And very often there's little things called bracts attached to them, which I'll come back to in a minute, but that's just to show you the arrangement to start with. In a normal flower, like I showed you last week, at the base of the flower stalk, you get sometimes you get a bract, okay? And those bracts can be very helpful in identifying different species from each other. And in the case of the carrot family, you might get bracts here. You might get them on these stalks, the top of these stalks, where you get little clusters. They're called bracteoles. If you get "ole" on the front of the word, it means mini or little. You know, it's a diminutive term. So if there's bracts present, they're here. The bracteoles might be at the base of these little mini rays, because that's what's called a compound umbel. So you can see here, this is an example of, I think it's cow parsley. I took these photographs last year and I stupidly didn't label everything, and I can tell most of them, but I think this is Anthriscus sylvestris, the common, is it cow parsley? Because it's got bigger petals on the outside of the ray. Anyway, so you've got a bracteole, a bract here at the base, and then at the base of each of these mini rays here, you've got the bracteoles, so bract and bracteole. So this is the part of the umbrella with a cluster of flower heads at the top on their own little rays, and you've got bracteoles here and bracts there. And that's two examples showing you there to show you bracts at the base, bracts there, like this little diagram. So that's sweet Sicily. Now the hogweed, which is what this is, is supposed to have no bracts here and bracteoles here, but I actually can't see any. I've got to be honest. I've checked all the botany books and all say that there should be bracteoles at the base of these little clusters here, and I just can't see them. So anyway, that's just to show you some variation. Anyway, you can quite clearly see the bracts there, the bracteoles there on that one. And that's just the back of the ray, looking at it from, you know, looking down onto the cluster. So that's the top of the bracteole looking down from above. Now, this family can be quite difficult to identify because there's so many white flowering umbels, and they do look very similar unless you get your eye in and start to see what the differences are. So the kind of thing you look for is a degree of hairiness. I mean, hogweed is really quite a hairy plant. And the shaping and lobing of the leaves, especially the lower leaves. The stems are usually hollow, but occasionally they're not. Whether there's bracts and bracteoles present on the flowers. If they are present, how many? But most of all, the ripe fruits and the pattern of the ridging. That's very important in identifying, very important. Oh, what am I doing here? Sorry, I've gone pressing the wrong button. Very important in identifying the different species. So there's cow parsley, and it's got very highly divided leaves. That's one thing that, you know, straight away that gives you a clue that this particular species, the leaves are very divided, very pinatified, and you know, the flowers will give you more clues as well. So you have to look at a combination of characters to work out what each species is. So let's look at the flower structure. And again, the whole lot of the flower is condensed into these tiny little structures. So this is pig nut, and here's the little rings of flowers, and the anthers, the stamens coming out here, 5 stamens, 5 petals, and 2 stigmas, which that looks like possibly 2 there. It just gives you the general idea of this, how small these flowers are. And each of those is a mini flower containing all of the things that, you know, typical diagram I showed you earlier. 5 calyx teeth might be present or not. If they're there, they're just tiny, you know, sepals. 5 petals, 5 stamens, the ovaries underneath the petals, don't worry too much about that yet. And the stigmas, it's 2 stigmas there. Now the fruits, as I said to you, are really important. This is sweet Sicily, and you know, the ribbing of the fruits, you know, the ending of the fruit, very important for identifying the different species. That's sweet Sicily. And oh yes, something else I should have mentioned is not just the ribbing, but also are the fruits flattened? They can be flattened from top to bottom or side to side. And again, that helps you to identify which fruits. You really do need fruits when you're identifying a lot of these species. That's hogweed with the mature fruits. And that's a close-up showing them. And again, they've got very characteristic shape, like little, almost like wings around the side there. And quite different from the sweet Sicily I showed you earlier. Another important characteristic of this carrot family, as I've said, are the leaves and how they're arranged. And you can see some of them, that's the, looks like pig nut there, which has got very divided leaves. And then here, compound leaves at the bottom, where there's the main leaf stalk. They're divided once, they're divided again, and then they're partly divided again. This one is lobe, this is wood sanicle. So that's not that very differently from that one. And the sheathing base, this is where the leaves are emerging out as the plant grows. That is really quite diagnostic for some species, and it's very characteristic. Some kind of sheath, like a sort of membrane around the developing leaf and flower bud, is quite characteristic for this family. So, you know, you look at the leaves, you look at the fruits, you look at the flowers. The stems, again, there's the spotty hemlock. That's a hairy stem of hogweed. And that's, I think that's just hogweed showing the hollow middle. So the degree of hairiness, or are they glabrous? Have they got no hairs on at all? All of these things will help you when you're looking. So all we're trying to say at the minute is there's lots of things to think about when you're identifying a species. And you're not going to remember them all. It's just that when you start to look at the plant, you'll think, well, yes, is it hairy or is it smooth? You know, are the leaves branching? Are they entire? And those kind of things they'll become habit as you start to observe plant species. They become second nature to you, and you start to look at the things which help you to identify one species from another in the same family. And those features are called diagnostic features. And there's just a few photographs. That's a giant hogweed from somewhere up on the Scottish Islands that Geoffrey Chater took. You know, they have hemlock. There's some sweet Sicily. There's wood sanicle. And I just thought I'd finish with this one because it's this is the Strantia, which is in the same family as the wood sanicle, but I didn't have any photographs of the sanicle, and I wouldn't want to pull a wildflower apart, but this is related to it. It's not exactly the same. So this is, you know, the masterwort or a Strantia that you grow in the gardens. I don't know what else it's called. It might have another name. You can see these flowers come out from a central ray, and they've got this very long case here. It looks like a sepal case. And there you can see all the stamens coming out, and looks like a stigma there. And that's a close-up there, for sure. So quite different from the flowers we looked at earlier. And, you know, don't at first sight look like a typical carrot family member. You know, the flower heads are really quite different from something like the pignut or the sweet Sicily we've been looking at. No, there's not an umbrella in the same way. I've just said thank you to the photographers at the end because I never do. And that's it for tonight. So, Margaret, do you want to add anything about the APAC, about the carrot family?
I've just put a message on chat about the fruits.
Go on, yeah. Would you like to explain?
Well, oh, stop that, please.
Sorry, I was just trying to find a diagram for you.
The fruits you've already mentioned can be long, wide, flat, spreading sideways or lengthways. Sweet Sicily is an excellent one, but it's much too early in the year to see it. But the fruits, and I can't just remember the structure of the ovary. Does the ovary have 2 ovules?
I think it does. I think it's got 2. Just check.
Right. Lizzie doesn't know either.
Yeah, I'm just checking.
The ovary, there's a carpel really, the whole lot. The carpel splits into 2, and they are attached at the distal end. That's the end away from the stalk. So that's something you might look out for when you see them. And very often, I'm trying to think of an example. Yes, hogweed. You all know the big hogweed, or even head parsley. Hogweed is a good example in that the fruits have got dark lines on them, a pair of dark lines. I can't remember whether they're characteristic or not at species level, but they are part of the structure of the fruit. Or it's something you know you could look out for. Another thing that is that in many cases, when you're looking at an umbel and the flowers are all open, you notice that the petals, the 5 petals, are of different sizes. And if you look closely, you may find that in the center of the umbel, the petals are of even size. All 5 of them are of even size. But as you get to the outer part, the outer ones, the outer 3 are larger, and on something like hogweed, the ones around the outside of the umbel, each individual umbel will be larger, so it's almost mimicking a single flower head with ray florets on the outside, wide ones like daisy, and small ones in the middle. Have a look at them when they're in flower, because that combination is really beautiful, and the picture on the screen doesn't help at all.
Do you want me to get one up?
They are really very beautiful. It makes them very lace-like. And you might think a bit about the beauty of flowers as well as just the structure. Right, I think I've said enough. Someone else has a question.
Thank you. Yes, you're right. It's a 2-celled ovary. Any more questions? Okay.
Yes.
Sorry, if anybody's trying to get a question, my touch screen isn't working, so I can't see. So
Derek
can you
has one.
just speak? Yes, I know, Margaret. I'm just trying to explain to people. Derek, go on. Sorry.
Yes, can I ask Lizzie, on the Myrethes odoratus, Sweet Cicely, on the leaf you get a white marking that always looks as if it's a disease of some sort, but I'm sure it's not. It's more characteristic. But does this ever occur on any other plant? And can you elaborate a little bit on what it actually is? Do we know?
I don't know. I know what you mean. The white markings on the leaves.
Yes.
Yeah. I've never seen. I'm not aware of them on any other species of flower family, but I'm not an expert. Margaret might know, but I don't know what they're. I don't know whether it's the edges always. I can't really. I remember seeing it, but I can't really picture. You know, is it? Do you think it's all time in the season, or is it towards the end of the season, Derek, when you see it?
Mm.
No.
What do you?
No. Hello.
Hello, Margaret.
Sweet Cicely has got white markings on the leaves. It's just the pattern of colour on the leaves.
Okay.
It's not a seasonal thing that develops. It may be more noticeable because you notice larger leaves later in the season, but it is there all, from them first expanding.
Oh, well, thank you, Margaret. So it's just, thank you, Derek.
Thanks.
Anybody else got a question?
Yeah, Laurie has asked a question. She asked if it would consider it okay to identify an umbellifer at a juvenile age, i.e. like now.
Now?
Yes. Can you identify an umbellifer when it's juvenile?
Yeah. I'm relating to something I did this weekend.
Well, I suppose it depends on which one it is, really. I mean, some because the basal leaves are important when you're identifying this family. So you get things like hogweed, but some of the others are quite confusing, I think, when they're young. Margaret, what do you think?
Can you hear me?
Yes, I can. I agree. Can you hear me?
Yes.
Yes. Not easy.
No.
No. I mean, something like Sweet Cicely, if you crush the leaf, it may smell of aniseed. I dare say some of the other umbels have also got characteristic smell. Whether very young plants will give you that smell, I don't know. I've got plenty of leaves of Sweet Cicely just coming up in my garden, but they're not seedlings or juvenile plants.
Okay.
Difficult. I wouldn't try.
No. The other thing is, it might be something, that you shouldn't be, if it's poison, one of the poisonous ones. If you're, it's best to be careful, really, as well. I guess if you're not sure.
Yeah, I agree. I was just looking at poisonous hemlock this weekend alongside a river, but then I realised it could be water dropwort, and I was thinking, could I distinguish a difference? But they were so, they were barely 6 inches tall, and it was very difficult to define what the differences were. And I was just interested to know. I could identify hogweed from its leaf pattern, but some of the others I don't know until it flowers and everything else yet.
No, well, I think we all agree with you. It's just too difficult, isn't it? A lot of them. Okay. So yeah.
Can I come back there?
Yes.
Hello.
Yes, Margaret.
Lizzie, you mentioned that hemlock, poisonous hemlock, had got purple blotches on the leaves.
Yes.
I think it's useful to know that. Sure, I was just having to think about it. My brain didn't work as fast as it did. Hedge parsley. Hemlock has got a smooth stem without hairs, a glaber smooth stem with purple blotches. Hogweed has got a rib stem which is very, I think it's very shortly hairy, but doesn't have any purple on it. But hedge parsley has got it's got a rough stem that's ridged and just have purple stripes on it.
Right.
Okay.
Yeah, thank you.
Long time ago, I remember doing something for a weekend course on roadside verge plants. I'd have to think hard. I'll try and do it for next week for you, of the ways of distinguishing common hedgerow umbels. One of them, we've talked about hedge parsley. There is an upright hedge parsley. It's an annual. It appears on the roadside verges in the sort of August time, the late summer, and it grows. It is very upright. It's almost as if it's in a straitjacket. It looks like a rather pretty little cow parsley. Everything's a bit smaller. It's pinky mauve in colour, and the stems are decidedly rough.
Thank you.
That's a nice thing to look out for towards the, towards the end of summer, when a lot of things have finished flowering. It's just starting.
That's true. That's a good thing to think about. So that's lots of interesting points you've raised there, Margaret. Thank you. Any more questions to ask Margaret when she's in full flow here? Anything else you want to query? Okay. Can you stop sharing then, Tricia? Please. I think we've got.
I can try.
Oh, yes. No, it's just that I think Naomi wants to mention something if we finish talking about. Oh, here we are.
I have a problem. My touch screen isn't working.
Okay.
So it's going on strike. So we seem to have 4. I don't know. Have we got 4 questions in? There's a 4 in the chat area. Is there something in the chat area?
I think we've done that. Naomi wants to speak about something else in a minute.
Right.
I think we've covered everything else, haven't we? Any more final questions before we
hand it over to Naomi?
Can I ask something? Could I ask something? Just yeah.
Before you go, sorry, did I miss something there? Before you go, may I just remind you all, next week we've got Tom, chairman of the Teesdale Special Flora Trust, who will talk about the trust. I hope all of you be present, plus those other Zoom attendees who are joining in on the blind identification. And I hope that you have filled in your membership form if you want to join the feed meetings, which will start on the 9th of May.
Thank you, Margaret. Judith, you've got a question?
Yeah, just going back to the daisy family. Is that okay?
Yeah, yeah.
Just the very last species you mentioned, the yarrow.
Yes.
How do you know that the outer white parts are florets rather than petals?
Well, sometimes they're just they're sterile in some flowers. I don't know whether they are in the the milfoil family, but if you take the flower off, you know how I was dissecting the flowers and showing you how they looked. Well, if you just take the flower apart, you'll see whether it is or not. They're quite tiny to open up, but in some flowers, in some species, the outer ring are sterile. So
does that make them a floret?
Makes them a sterile floret, I suppose.
Okay.
It's a good question, and I forgot to mention about sterile. You can get sterile rays. I think daisies can sometimes. Some of the daisies, Margaret, can we have sterile ray? Can we have sterile ray florets in some of the daisy family?
What's the situation in some of the centurias that have got very big outer florets?
Yeah, I think some of, I don't know.
You get them in the south of the country, Jakia.
I'm looking. Yeah.
In the meantime, Judith, is Judith listening?
Yes,
Yeah, Judith, I wonder if I misled you a bit, but caused a little confusion when I talked about the outer florets in hogweed having larger outer petals than inner petals. So you've got 3 large and 2 small, or it may be 2 large and 3 small, because the petals are divided almost into 2. But if you were to take one of those florets off, you'll be able to see that if you try to take one of the petals off, you would find it was attached to the other 5 of the 4. All right?
Yeah, thank you.
Thank you.
Or have I confused you more?
No, that's fine.
Yeah. Thanks, Margaret. Yeah, I think as far as the milfoil, the achilles are concerned, I would just get a flower and have a look. I just didn't dissect them open last year. I didn't think about it, and so if you pull one off, you might have a tiny ovary at the base.
Okay.
I can't be certain because I didn't do it, but and it says that they've got ligulate flowers, and this is Stace, who that's what we use to check everything. The new flora of the British Isles. But I know that some of the daisy family do have sterile florets. I just can't think offhand which ones it is. So thanks for that question, Judith. I'm glad you reminded me, even though I can't answer it properly.
No, it's okay. So does the yarrow not have any petals then?
No, the yarrow has tiny flowers.
Yeah.
I can't bring it back up. It's got tiny flowers.
Yeah.
Of 2 types: ligulate flowers on the outside and tubular flowers on the inside. But because they're so small, they only have like 4 or 5 tubular flowers, and then a little ring of ligulate flowers on the outside.
Right.
Yeah. Mm-hmm. But they're tiny. Yeah. So it's because we've been doing 2 families. So I'm going back to the daisies now. You see the daisy family.
Yeah. Okay.
Yeah. So if you have a look at them, I'll send you a photograph if you want as well. And you can have you email me.
Thank you.
Anything else from anybody?
Lizzie, I think the outer florets of Centuria are sterile.
They are sterile. That's what Mark said.
Do
you want to say that again, Anne? You've gone onto mute.
Sorry, I'm just thinking that I think it's the outer florets of the Centuria that are sterile.
That's what Margaret said.
All right, yeah.
Yeah, that's great then. Margaret, you're right.
Thank you.
I think Margaret probably knows better than me.
The outer florets of Centauria are sterile. Anne's just looked it up.
Is that so for all Centuria species?
I don't think so. I looked at
These are the knapweeds, folk.
I don't think knapweed is. I don't know. We'd have to look that up. Yeah.
There's a knapweed, an upweed that is more southern than this part of the country.
Maybe. We'd have to check.
Centuria,
Oh, I know which one you mean, yes.
Here.
Yeah, I know which one you mean. That does look as if it's got sterile outer petals, doesn't it? Right. Well, thank you, everybody, then. So that's the daisy family and the carrot family done. So when you go out for the next, I've actually sent you some homework. What I've done, just to finish off, I should have said this, was in the notes on the daisy family at the very end, I've shown you an example of how to try and work through a particular species and identify like a particular group, like the genus, and work out the different species inside it. And I've given you some examples to try. It's a bit early yet because there's very little out, especially if you live in Annenheads, when everything has gone back under the ground again after the cold weather today. But it might be a good way to help you to get your head around, looking at the different species in the same genus. So if you want to do that, it's on that PDF that we've sent round. If you've got any problems with it, just let us know and I'll resend it. Okay.
Is that time for me now then?
Oh yes, sorry, where are you? Sorry, Naomi.
Thanks, Lizzie. I've just had some colt's foot just appeared in my vegetable patch, so I shall look at it with renewed respect. I just wanted to grab everybody's attention just for the last few minutes today, just to mention that Plants on the Edge has been fortunate to get some funding for first aid. So we've arranged a course on Wednesday, the 20th of April at UTAS called Outdoor First Aid Emergency Action. So it'll be all day and we have got some spaces left. So it would be really good if anybody would like to come along. There's no cost to you at all, but these places have been paid for. So it'd be really helpful for us to fill those spaces. I think I've just put my email address in the chat and I haven't sent it yet. I will send it now. My email address is there, or you can contact me on Facebook, or hopefully go through Tricia if anybody can't get me any other way. So that's Wednesday, the 20th of April at UTAS, first aid course for anybody in the botany group.
That's brilliant, thank you.
What can I ask? What time? Sorry, what time does it start? What is it?
It's all day. I think it's a 10 a.m. start, but I would just need to confirm that.
Yeah, I think yes.
Yeah.
I think it's 10 o'clock. Yes.
Okay. Thank you, Margaret. Does anybody have any other questions about first aid? Great. Well, if you do, if you email me or Facebook or call me, anything. Yeah, thank you.
Thanks, Naomi, and get well.
Thank you.
Right.
All right. Shall I close the meeting?
Yes, please, Margaret. Thank you.
At least I should be able to close the meeting, even if I couldn't start it. Yes, I think, Lizzie, I think you've done a very good job of introducing plant identification. I hope everybody's found it useful and that I do know at least one person was stimulated to go and look at Ranunculacea in their garden after last week. So yes, coltsfoot are still out, dandelions are out. I haven't seen any umbelliferous flowers yet. So you better have a go at the forgotten its modern name now, the composite family.
Asteraceae.
And try and get a start. If you've got questions between now and the beginning of May, if you like to send them to me, if you've got my email address, it's been published enough. I'll see what I can do about answering them. If not, I shall send them on to Lizzie.
There won't be many coming my way if you're answering them, Margaret.
All right, thank you very much indeed, Lizzie. We'll unmute ourselves, say thank you. Good night, all.
Thank you, Margaret. Thanks, everybody. Thanks,
Thank
Tricia.
you.
Bye, everybody.
Thank you,
Bye.
Lizzie. Thank you.
See you in the field, I hope.
Yay.